obviously, the next question then becomes, how do we get the translation of that coding mechanism in DNA? How does it transmit that coding into proteins? We know that DNA codes for proteins, but back then they didn't know how this process worked. It basically was discovered pretty quickly that RNA acts as a intermediary. So here you can see the DNA in this picture. This is called transcription. The DNA is in the nucleus, and that DNA then is transcribed into something called messenger RNA. You can see the mRNA there. Now DNA never leaves the nucleus, so it's protected by the nucleus. That coding system never has to get out and interfere with other processes happening outside the nucleus. It's the messenger RNA which goes through the wall, which is able to go through the wall of the nucleus and go out into the cytoplasm. So this is transcription. It's preserving the DNA in the nucleus, but it's taking the messages out through this messenger RNA, and it's carried out into the cytoplasm. It's transported in a way. And then when it gets out there, it goes through a translation process. Now, so let's talk a little bit about RNA. We haven't talked much about RNA before. RNA is very similar to DNA, where DNA is a deoxyribonucleic acid. RNA is only a one ribose, so you get only one ribose there, not a deoxyribose, and also you get 4 bases. So ribose and the 4 bases, as you do in DNA, but one of those bases is different. You have cytosine, guanine, and adenine, but you don't have thymine. That's because it's replaced by uracil. So the only difference in RNA is the ribose versus the deoxyribose, and the uracil versus the thymine in DNA. So a little bit different, but enough to where it impacts everything. So let's go now and take a look at this process of transcription that we've been talking about in a little more detail in the nucleus. This is a blow-up of a chromosome, and what we've done on this chromosome is we have one gene there in the green, and the idea here is to take that gene and convert it to messenger RNA, and I want to show you how this works along the chromosome. So what happens is we have a promoter. You can see the promoter there at the beginning of the gene. So this tells the reading process that you're supposed to start here with the gene and then start coding it, taking the DNA and converting it into messenger RNA. And you can see that the DNA here, which is T, C, T, A, G, etc., is going to be converted to this messenger RNA. There's going to be an A that matches the T. Remember, we have A's and T's and G's and C's matching up. And then you have a C, you're going to have a G in the RNA, and then a T is going to be an A. And then look what happens next. You have an A, but rather than have that a thymine in the RNA match up, it's going to be a uracil. So that's basically the difference. But everything's the same. It's basically translating the code of the DNA. And then eventually you'll get to what we call a transcriptional terminator, and this tells the whole system to stop making mRNA. So it's really a beautiful system. Then you can not hurt the DNA. You're just decoding one side of the DNA in the first place, and you're creating a messenger RNA, and then this messenger RNA can be taken out through the nucleus, and it can go out and it can make the requisite protein that this gene has been coded for. We'll talk more about that in a minute. I also want to bring to your attention the regulatory sequence over there on the left. We didn't know what these regulatory sequences were for the longest time, but basically the regulatory sequences, these are these are things that tell the cells, or excuse me, the genes, when to turn on and when to turn off. So let's take, for example, stem cells. I think you all know that stem cells can become any other type of cell. So when a fetus is growing, there will be some regulatory sequences in there that will tell the cell, oh, you need to become a liver cell, or you need to become a bladder cell, or a skin cell. And so these regulatory genes then help the process of the growth of various types of organisms in the fetus. Now, it's not completely understand gene regulation yet, but we're beginning to understand it more and more all the time. And it's really one of the reasons why we've discovered that there are so few genes. There are only about 20,500 genes in the DNA. And so we've got this funny situation where the regulatory sequence becomes as important as the genes. We thought we'd have many, many more genes than this, but that regulatory system, which is turning on and off the genes and telling them when to go do their thing and convert to something, to a protein, well, that's all part of this regulation. So gene regulation becomes very, very important. All right, now we've been talking about transcription, but I'd like to now talk about translation. And that's when we have the messenger RNA out in the cytoplasm. Then the cell has to take that messenger RNA and it has to start making proteins from the messenger RNA. So let's talk about how that happens. And I want to first of all talk about Crick for a minute. Back when Watson and Crick discovered the structure of DNA, Crick postulated that the coding system would be in bases of three. Three bases would determine the amino acid. Now we know that there are 20 amino acids that go together to form the proteins, and of course the ultimate goal of these genes for the most part is to form proteins. So the amino acids have to come together to form proteins. So Crick reasoned that the coding mechanism was in groups of three that told the system what amino acid was needed. So three bases coded for an amino acid. And Crick reasoned that because you can have 46 different ways of matching up all these bases, the bases that we have to make 20 amino acids. If you use threes. If you have four bases to do coding, you get way too many, and if you only have two, then you don't get enough to cover the 20 amino acids that are needed. Now obviously with 46 different computations of arranging three bases, you've got more than you need, and we're going to find in a minute that you do have coding of different bases for the same amino acid. And in the early going, by the way, the three bases now are called codons, and these codons then are actually coding for amino acids, and this is called basically the genetic code. Now, in this diagram, this is called a lookup table, and just to show you here, we're dealing with glycine. So you start out in the middle of the circle there, and you'll see G, and then the next one out is G, and then you have four letters there. They're GGU, GGC, GGA, and GGG. So all four of those combinations of codons will give you glycine. They code for glycine. So this is how we look up what bases code for which amino acid, glycine being an amino acid, the codons GGU, GGC, and etc. Those are the bases that come together that code for those amino acids. And Crick figured that out way before they determined that the bases came in threes. He postulated that it was three. Kind of an interesting side note on the whole system. Well, let's take a look at this graph. The picture of Marshall Nirenberg, Mattei, and Khurana here. And what Nirenberg and Mattei did was they said, okay, how are we going to figure out what these bases code for? they realized that they came in bases of 3. So they said, well, the easiest way we can do this is just make an RNA full of uracil, only uracil, and we'll see what protein gets coded for that. So they managed to create a sequence, all uracil in the RNA, and then they found out that that produced phenylalanine. So phenylalanine was the product of having 3 uracils. You can look it up on that table. And then Khurana got involved, and they figured out CCC and GGG and AAA. So they began then through this kind of monotonous way of programming the DNA. They started to figure out what bases coded for what amino acids, and it turns out Nuremberg and Khurana won the Nobel Prize for their work. Unfortunately, Matai died, so he didn't win the Nobel Prize. Again, they didn't give it posthumously, unfortunately. And they won the Nobel Prize in 1968. So let's take a look now at translation in some more detail. Remember, transcription is converting the DNA to messenger RNA in the nucleus so that the messenger RNA can leave the nucleus, go out into the cytoplasm. Once it gets out into the cytoplasm now, it's going to be picked up by the ribosome. Ribosome is a biological machinery that literally reads the messenger RNA. And it reads the messenger RNA from left to right. And what happens is that it has these transfer RNAs. They're called tRNAs here. And they match up to the coding system in the messenger RNA in 3s. So you have a tRNA with, say, UUU, and it's going to go out and it's going to find lysine, which is the amino acid that it codes for. I also told you it was phenylalanine. Well, it codes for more than one. And it's going to go out and it's going to find lysine, and then it's going to bring it back, it's going to set up, and then you're going to have a whole series of these transfer RNA bringing in the amino acids, and these amino acids then are going to form a growing peptide chain which will eventually create these proteins being made up of amino acids. And of course different proteins are created by different sequences of amino acids. So really this is the whole process going on out there in the cell to create proteins. So we have now, through translation and transcription, we have the whole process of what we call the central dogma, which is you start out with DNA, which goes to RNA, which goes to proteins.